Okay. So anyway, I'd like to thank you all for the opportunity to being here. I've given many talks, but I've never given a talk in a prettier venue than this one. So most of you probably expect to hear me give a talk about the work that led to my prize, but I don't want to do that for two reasons. One, WE did a really good job of explaining it, and two, well, three reasons. Two is that you can find various versions of that talk online already, including the Nobel lecture. And three, although I really love the story of developing the palm in Harold's living room and working with it for a number of years, I'm so sick of telling that story over and over and over and over again. So I'd rather tell you about a little bit more of what we're doing now. So there's many problems with super resolution microscopy, but in a live imaging context, one of the fundamental problems is that regardless of which method you decide to use, getting higher and higher resolution requires more and more pixels, which of course means you need to take more and more measurements, and that takes more and more time, and also means that you're throwing more and more damaging light at your specimen. So if you're going to be honest about it, there's always trade-offs between spatial resolution, temporal resolution, phototoxicity, and it gets harder as you go deeper. And so around 2008, when I was aware of those limitations, I got really bored of super resolution. I started moving in other directions. And so the guy who really, I think, appreciated these challenges better than any of the rest of us in the super resolution field was Swedish native Mats Gustafsson. And so he developed yet another method of super resolution called structured illumination microscopy, which does not require the switching principle that the rest of us use, but instead uses patterned light to create beating patterns of the structure in the sample against that light pattern, and then works in Fourier space to then reconstruct an image at up to twice beyond the diffraction limit. Now, one of the reasons that that technique has not gotten the attention it deserves is everybody says, oh, well, if Stead and Palm can do 10 times, what's the big deal about 2 times? Well, the big deal is that it's much faster and uses much lower intensities, and Mats appreciated that. This is a great example showing the endoplasmic reticulum. Now, you've seen a little bit of live imaging by other methods, but you've never seen something going for 1,800 time points at sub-second rates. And so you start to appreciate of what that time dimension brings to the table in terms of our understanding of how things work. Because the thing that defines life is that it is animate and dynamic. And SIM is a great tool to be able to see that. I think Mats's crowning achievement, though, was in figuring out how to use 3 beams to create SIM in 3 dimensions, so that you can get 2x beyond the diffraction limit in x, y, and z. In one example, my group, sadly, Mats died of a brain tumor in 2011 after I recruited him to Geneva in 2008. So in a way, Mats is the Messiah. I have become John the Baptist and sort of an acolyte trying to, I love localization, but at the same time, I love SIM, and I now consider it my calling to talk about the virtues of SIM as much as the virtues of localization. So in one example in my group now, we've used the 3D SIM with Jennifer Lippincott-Schwartz to study the forces that are involved in cell motility. In this case, the green represents actin or myosin head groups, and the purple is the actin which is bound together by these head groups, which create forces to move the cell. And by the 3D imaging, we're able to show that those forces aren't down at the bottom of the cover slip where you would think, but are actually on the top and squeeze the cell to contract it as it goes forward. But honestly, when we've had many, many groups come to our lab to use all of our microscopes, in the SIM scope, what most people have gravitated towards is the turf mode, where you just see the very bottom of the cell because it's fast and because you're not limited by out-of-focus fluorescence. And so this is an example of looking at the immunological synapse of a T cell presented against an antigen-presenting cover slip. You can take up to 10 frames per second, so you can look very quickly. We've used that with a group at NIH to show that myosin 2, which has 3 different isoforms, there was a controversy in the field about whether these isoforms could mix in these bundles of myosin filaments, and we showed that they could. And then finally, another nice thing about TURF is that you're not limited by the out-of-focus fluorescence. So here we plop down a whole Drosophila embryo, and you're looking at these amniocerosis cells in early development, and you can see the single actin filaments going through these pulsating contraction expansion phases of positive feedback oscillations that drive these wave-like motions of those cells. So if the knock against SIM has been that it only has 100 nanometer resolution, what can we do about it? Well, the easiest and stupidest thing is not to apply the fancy tricks we've heard about already, but just increase the numerical aperture of the lens. So Olympus sells a 1.7 NA objective, which takes you down to 80 nanometer resolution, which is already in the ballpark of a lot of the data that you can get by other methods. With that, we then looked at clathrin-mediated endocytosis, or how things are brought into the cell. So normally these clathrin-coated pits form these sort of domes like this. Because the evanescent field is so short, you're only seeing the top, so they start to look like rings as we look at them then at this 80 nanometer resolution. But rather than just looking at them statically, you can look at them dynamically for a long period of time, and you can follow the entire course of the formation of these pits, their motion, their growth, and their eventual pinching off and internalization inside of the cell, again at high speed for many time points. Another virtue of SIM is that it can use any standard label. You don't need any tricks of the labels, so it's very easy to do multicolor imaging. So here you're seeing the cortical actin, as well as its relationship then to these clathrin-coated pits. And you can see 2 types of structures, individual rings for individual pits, as well as these larger plaques that are aggregations of pits. So we've looked at that carefully, and we've seen there's another controversy in the field about what is the role of actin in clathrin-mediated endocytosis. And so what we've found is about half the time clathrin is involved and half the time, actin is involved, and half the time it's not. But when it is involved, the lifetime of the pit is shrunk significantly. In addition, these plaques we've been able to show there's some models and some EM data that suggest they're just flat lattices of clathrin, but at least in these cells, they're nothing more than aggregations of pits, and sometimes individual pits spin out of them. And furthermore, a real surprise was finding these actin rings that are like clathrin rings. When we first saw them, we thought, oh, well, that's helping to pinch off the clathrin. But they're completely unassociated with the clathrin, and their role is completely unknown at this point. So that can get us to 80 nanometers without using any tricks. But what if that isn't enough? Well, then we need to go to the sort of switching mechanisms that Stefan and WE discussed. This is something that's long been known to both Mats and the other pioneer of SIM, Rainer Heinzmann. And so Rainer suggested back in 2002, using various types of saturation or other means of getting nonlinearities, such as the depletion that's used instead in order to get high resolution. Mats demonstrated that in 2005, and then in 2012, after he passed, his grad student Hesper Rigo was able to use the photoswitching of fluorescent proteins like WE described, and then the sort of depletion, but now depletion with a grading structure, in order to be able to get additional harmonics to take you from the 2x resolution to, say, 3x resolution that gets you down to 60 nanometers. And so we've started to use that method. The problem with this demonstration was it took forever, 900 seconds to get one image, way too slow to do live imaging. So we've pushed that in our group. This is now doing half a second acquisition. This is looking at the actin cytoskeleton as you go from linear to nonlinear, and before that, just regular TURF and deconvolution, and then we can do live imaging of the actin cytoskeleton then at 60 nanometer resolution in this case. We've then applied that in a variety of systems. This is a particular case where we're looking at that actin cytoskeleton in relationship to Rab5A, which is a marker of early endosomes. And so then we can study the relationship of how these endosomes track or diffuse with respect to the cytoskeleton. And what we've been able to see see is a number of things. Unlike the clathrin-coated pits, which have a very stereotypical round shape to them, those rings, the early endosomes have crazy irregular shapes. So I picked 3 examples here, and I picked them carefully to match an EM paper that appeared 20 years previously, but you can see that we can now then start to see these shapes. And for example, these little protrusions are bits of membrane that will eventually recycle back to the plasma membrane. And furthermore, we can see in these endosomes that there's little dark holes often, which probably represent either a cargo or vacuole inside of them. And oftentimes you'll see them, normally they diffuse sort of randomly in the membrane, sort of constrained by the actin, but then sort of next to but not on the actin, you'll see whoop, one go just real fast out of the way, and that's probably microtubule active transport of these early endosomes. So the moral of the story is that by getting away from that myopic focus on spatial resolution, and backing off a bit, we've been able to open a whole new application space that the extreme high resolution methods have not yet achieved. And so if that's the case, it sort of begs the question, well, what if we back off further? What if we go to the diffraction limit itself? Can we make a microscope that would be as much of an improvement over existing live imaging tools in terms of its performance for temporal resolution and non-invasiveness as the STED and Palm and so forth have been an improvement for spatial resolution? So when I got bored in 2008 of super resolution, that's the goal I set for myself. So the good news is the standard live imaging tools leave a lot of room for improvement. They're basically the wide field and confocal microscope. The problem with using these in a 3-dimensional context is they're used in this epi configuration, where you illuminate the whole sample and the light comes back. But there's only one plane that's in focus at a time. So all you're doing is creating out of focus background and doing premature bleaching and toxicity of the rest of that sample. In my opinion, a innovation in microscopy that appeared 10 years ago that is at least as important in super resolution is when Stefan's old PhD advisor, Ernst Stelzer, reintroduced the 100-year-old idea of plane illumination. In this case, instead of bringing the light from above, you take a second lens and create a sheet of light from the side. And so therefore, only the plane that's in focus is illuminated. So there's no problem with bleaching above and below. And furthermore, because the whole plane is illuminated at once, intensities are low, you can use a fast camera to catch a whole plane at once, and it's very fast. So this method has been transformative for understanding embryonic development at single-cell resolution. But it has one problem, and that's that, again, diffraction and the laws of physics place a trade-off between how thick that light sheet is and how big of a field of view you can look at. And if you're going to apply it to the size of a single cell, it's going to be on the order of 3 to 5 microns thick, which is on the order of the thickness of a single cell, so you don't gain much benefit. So the trick that I and others brought to the table is rather than using Gaussian beams, there's a different kind of beam you can create called a Bessel beam, where you're able to, by using annular illumination in the rear pupil, decouple the length of this spear of light from its thickness. You can then sweep that rapidly in and out of the plane to be able to virtually illuminate one plane, and then go up and up one plane at a time through the specimen in order to see the whole thing. So that sounds like a slow process, but in our modern implementations, we do 1,000 planes a second, or up to 5 volumes per second through single cells. Here's a couple early examples where we're looking at the initial stage of gastrulation in C. elegans embryo, and with our collaborators, we're able to show the myosin in green, which represents those actin myosin contractions. They generate the forces, but the forces occur before the cells gastrulate. And so there has to be a yet as known clutch mechanism which couples those forces to the membranes. In another example, we looked at mouse embryonic stem cells, which are very light sensitive, even in a wide field microscope. But by using the low toxicity of this, we were able to show that those little beads that you see, they're impregnated with the signaling protein Wnt5a. And we were able to show that the gradients of that protein serve to orient the mitotic spindle such that the daughter cell, which is closest to the source, remains a stem cell, and the one further away becomes a differentiated cell. So there's a problem with the Bessel beam approach, though, in that it's not just that little skinny pencil of light. In cross-section, it actually looks like a bullseye, and it has all of these side lobes. So in our case, we came up with two solutions to address that. The first is to use something called two-photon excitation, where only the most intense light creates fluorescence. And so then the center beam dominates, and then in this case, we're able to very quickly scan volumes of filopodia on a HeLa cell to look at the dynamics of those structures. The other solution we used was to, instead of moving that beam continuously and sweeping it to get the plane, stepping it in discrete points across the plane to create a grating, and then we could use Matz's idea of structured illumination to sort of computationally strip out the effective side lobes, and at the same time poke the resolution a bit beyond the diffraction limit in both the X and Z directions. In order to do that, it took too much time to physically step the beam, so we put in a diffractive element that created an array of 7 parallel Bessel beams. That solved the problem of the speed, but what was shocking to us was how much less toxic it was to have 7 beams running at once instead of 1. And so in 5 years now of doing live cell imaging, what I've learned is that while the total dose of light you throw at your sample is an important metric of the health of the cell, a far more important metric is the instantaneous power that you deliver. So, seen in that context, point scanning, such as a confocal microscope, may be the absolute stupidest possible way to do live cell imaging. Because not only do you have this cone of light above and below the focus, creating all of this photo damage and toxicity, at the focus itself you have this actinic spot of light that's going and leaving death and destruction in its wake as it goes along. So the moral of the story is the genius of Matz again and the genius of Ernst, you need to spread the light out to keep the peak power low. And so if 7 beams works better, why stop at 7? So we started to model what would happen as you have more and more Bessel beams come together. The problem is that their side lobes then start to interact, so you have to model what happens in terms of the interference of these beams. And what we found is that there are certain magic periods where you get destructive interference of those side lobes to get rid of that problem, but you also have perfect 100% modulation in the plane for doing structured illumination, and you spread the energy out. So in a way, it's a triple win all at once to do this. As it turns out, these magic periods are related to a theory about optical lattices that I was trying to pitch and work on before we dropped it in order to do Palm. And so I dusted off my old theory about that and then applied that to light sheet microscopy, and that's what we now call lattice light sheet microscopy. So there's 2 modes in which you can operate this microscope. Either you can use that structure, take multiple images per plane, and then you get slightly beyond the diffraction limit, as you can see here, and you can see these filopodia are easier to see than here. Or you can just dither that structured pattern back and forth to mimic a continuous plane. Then you don't need to take 5 images per plane like you do here, but can just take 1, and then you can move like a bat out of hell through the cell. In this case, we're doing 750 volumes. For a protein which is highly expressed like here, the natural turnover and production of new fluorescent protein is faster than the rate at which we lose fluorescence due to photobleaching. And so you can look at a cell like that forever. You can watch it just divide and do its thing and never run out of being able to look at that in 3D at high speed. So one of the other advantages of this lattice light sheet is it's so thin that it's thinner than the depth of focus of a high NA objective lens. So that means one of the problems of single molecule techniques applied to biology is typically we've done this in a wide field context where the samples need to be really thin because otherwise the molecules that are out of focus create so much haze you never see the ones that are in focus. But here we're looking at a 35 micron spheroid of mouse embryonic stem cells, and by parking the light sheet at one plane, the signal to noise that you get there is as good as if this were a turf experiment. And so then you can bring the whole range of single molecule techniques to bear to do that. In this case, we were looking at transcription factors, in this case SOX2, and its transient binding to DNA to find regions of specific and nonspecific binding of those transcription factors in a paper that we published in Cell earlier this year. The other advantage is WE mentioned this paint idea. In the same year that we did PALM, Robin Hochstrasser's group said, wait, you don't need to photoactivate these molecules. They can be whizzing around in solution and just transiently bind, and when they bind, you can get them. The problem with doing paint is that the media is glowing and the media is thick, so it's hard to get good signal noise. So it's a perfect marriage with the lattice light sheet. The advantage of paint is that one of the major challenges of super-resolution fluorescence microscopy is getting enough goddamn labels on your sample that you decorate densely enough to get the resolution you want to get. It's a huge problem. And with paint, you have this infinite army of molecules in the media that can keep coming again and again and again. So in this case, this is looking at a whole dividing cell. You're looking at 300 million localization events to get that 15 micron tall cell into this level of resolution. That's about 2 orders of magnitude than a standard 3D palm experiment. And in this case, you can now then be able to image all of the intracellular membranes together, so it looks much more like an EM image than fields of dots or sparse samples, which is what you usually see in super resolution imaging. Okay, so, but the real beauty of the lattice light sheet is by spreading that light out and having a thin sheet is it's ideal for doing 3D dynamic imaging. So this is now looking at single cells. In this case, we're looking at cell division, looking at the chromosomes, and those crazy tracks are looking at EB1, a marker on the plus ends of growing microtubules. And so what we were able to show is that the velocities of microtubule growth increase as you go through metaphase and then slow back down again to cytokinesis. And this represents also the low phototoxicity because you're looking at 500 volumes in each of 2 different colors. Both volumes are collected in 1.2 seconds, and then so a total of 1,000 volumes from the same cell. It's going through all the mitotic checkpoints when the cell is super sensitive and would otherwise arrest and is perfectly happy. This is an example of looking at another light-sensitive specimen, Dictystelia, and showing an unexpected feature in actin, which is these sort of lightning bolts that you see here, which we now find common in Dictystelia. And finally, this is looking at a protozoan such as you'd see in a pond, Tetrahymena. He's trapped in a little pocket of agarose and frantically trying to get free, and so you can watch the beating of the cilia as he does that and get the mechanotransductive forces by looking at those. So now working from single cells to cell-cell interaction, this is our friend the T cell we saw in 2D before, but now we're looking at a T cell interacting with an antigen-presenting cell, and we've discovered this very fast flow of actin away from the immunological synapse along the boundary of the T cell. Here, much of what we know about cell motility is by looking at cells moving on cover slips, but really they move in a 3D matrix. So this is a collagen mesh, and working with Dyke Mullen's group, we're studying these neutrophilic cells doing 3D motility through these meshes to look at the differences compared to their 2D motility. So I probably clicked one too many times. Let me come back and see if I can get that to start. Okay, so at the largest scale, we can look at embryos. We can't look at big embryos through the whole thing, but we can look at the edges of them. Or in small embryos like early C. elegans, we can look at the whole thing. So here, Josh Bembenek, our collaborator, is interested in AIR2, which is a marker of the mid-body remnant that remains after cell division, those little blobs there. And he's interested in tracking those over time because he believes they have an ancillary role during development later on. In this case, remember I showed you the turf image of the Drosophila embryo going through development, and then there's the stage where the amniocerosis is closed up by the epithelium. This is now looking at in 3D with cadherins as these epithelial cells eventually encroach upon the rest of it. So the problem with extending any microscope technique from single cells into embryos or tissues is that these tissues are not uniform in their refractive index. So the light rays get scrambled, in this case the light sheet gets scrambled, and these meiotic chromosomes going in, and likewise on detection the light is scrambled so only the cells closest to the detection objective are clear. It's the same problem you have when you have water on your windshield and you haven't turned on your wipers. The good news is astronomers had to fix this problem 50 years ago, and they used a technology called adaptive optics. And so what you're able to do is actually measure that distorted wavefront, bounce it off a deformable mirror, change the shape of that mirror to exactly balance those aberrations, and get back to the diffraction limit. So you get huge gains in both signal and resolution by doing that. We've applied that in two-photon microscopy to look at the zebrafish brain in a live developing embryo 3 days post-fertilization. Here's with the adaptive optics on, going about 250 microns deep. That's turning the AO off. So normally when you're doing intravital or in vivo imaging at depth, you get that crappy resolution. And then by applying the adaptive optics, we can get back to the diffraction limit. That's not super resolution. That's just getting where you thought you were supposed to be. We can also apply it in a confocal context, which makes it easier to do multicellular imaging. So here you're looking at mitochondria that you can't yet see in the soma of a neuron 150 microns deep, but now we can do two-color imaging. Now we turn on the AO, and because we know the point spread function, we can do a deconvolution, and there now you see the mitos just as good as if this were a cell on a cover slip. And then likewise, here's the membrane around it. Here you're looking at oligodendrocytes, which are like the glial cells, and a wave of adaptive optics sweeping in and showing the improvement in the resolution and signal as we go along. So for the future, the goal of my group is to basically combine these methods. We normally look in cell biology at immortalized cells on cover slips, but they didn't evolve there. So we want to get looking at these things inside of whole embryos, apply the high speed and low toxicity of lattice light sheet, and then start scrolling in super resolution after that. But I'd just like to say, one of the key concerns I have about all of these new technologies is they don't mean squat if they're not used by biologists. In fact, we can talk about all the sort of physics tricks and so forth you want all day long, but none of it matters worth a hill of beans if you can't answer a question that could not be addressed by any other means prior to that. That's the only metric that matters. And to do that, these microscopes have to be broadly available. It takes time to do that. Ultimately, it takes commercialization to do the hard work to make turnkey instruments to make that happen. In order to get over that hump, my institute plus Gordon Moore Foundation has partnered to create an advanced imaging center at Genelia Farm. So in 2013, we had 30 different groups from across the world come through to use our instruments. If you just go to this website, you'll see more about that. And if you're interested in applying these techniques, you can come for a week or two at a time and then try to look at your own systems. And so with that, I'd like to conclude, and I've been blessed with many great collaborators over the years and really blessed with a great, hard-working group of postdocs who've really taken to heart the notion that nothing matters more than hard work in getting things done. I've also been blessed with a great group of collaborators, and I thank you for your time.